Again, I'm Sophia, and I'll simplify this a little bit. So we're just looking at sort of the existence of these 3 genes, which are found in Pseudomonas aeruginosa. So you'll understand. So just a little bit in the way of an introduction. In case you weren't aware, we have a bit of an antibiotic resistance crisis on our hands. Bacterial species, pathogenic bacterial species, are becoming extensively resistant to drugs at an alarming rate, which means that there's a really increased need for prudent drug use and to learn as much as we can about these drug-resistant strains. The escape pathogens are some of the most clinically relevant and extensively resistant species, and Pseudomonas aeruginosa is one of them. So it's a ubiquitous bacteria, which means that it's found in soil, water, et cetera, sort of all around you, actually. And it's gram-negative, and it's an opportunistic pathogen, which is why it's not making you sick when you encounter it all around you. It really only affects people whose immune systems are compromised or having these acute injuries or infections. It can infect a variety of environments, including the lungs, urinary tracts, and open wounds, but we'll be focusing particularly on its behavior in the lungs. And it's a leading cause of nosocomial infections, which is a really big problem for people with chronic conditions such as cystic fibrosis, who spend a lot of their time in hospitals. And the lungs of CF patients in particular are an ideal environment for Pseudomonas, so it's a big pathogen for them specifically. We'll talk about why that is later. They possess a variety of mechanisms of resistance, including the overexpression of efflux pumps that pump the medication out of the cell, the modification or loss of their porins, which stops the medication from getting into the cell in the first place, quorum sensing, which is a really interesting phenomenon, but we don't have time to talk about it now, and biofilms, which we'll define later. It is characterized as a high threat pathogen by the World Health Organization and other leading health organizations, so it is something that we need to look into. Finally, carbapenems. So we'll be talking about carbapenemases, which are enzymes that destroy this medication. And carbapenems are one of only 7 classes of antibiotics that can be used against P. aeruginosa and infections. And they work by inhibiting cell wall synthesis. And we'll be looking at these three carbapenemase genes and their associated proteins, BLA-VIM, BLA-SPM, and BLA-KPC. So, cystic fibrosis, just a little background. It's an autosomal recessive condition that causes mutations in that gene, which causes defects in this ion channel, which controls salt transport across epithelial cells. When salt can't be transported effectively, we get this highly viscous sputum, which can't be cleared properly from the lungs. And this is actually really good for P. aeruginosa because of the way that it replicates. So it can spend its first phase of rapid growth feasting on this extra nutritious mucus, which is full of stuff that they like to eat. And then, of course, once they're big and healthy and strong from all those nutrients, they can move and continue to grow and destroy. So like we said, this is an ideal environment for them in CF lungs. Biofilms. These are colonies of microorganisms, bacteria here, but they can be other kinds of organisms as well, that are encased in this slimy, sticky protective matrix that they produce themselves. They can do a number of things that lead to persistent infections, such as slowing the absorption of antibiotics, allowing for increased horizontal gene transfer, which spreads a lot of antibiotic resistance genes, and harbor persisters, which are these bacteria that can enter like a dormant state and ride out a course of antibiotic treatment. P. aeruginosa biofilms in particular can inactivate the complement system, which is sort of the immune system's first line of defense, using these proteolytic enzymes. And they can also switch their metabolism to fermentation if need be, which allows them to survive anaerobic conditions in CF lungs. So the first thing that I needed to do for my investigation was retrieve the files of these 3 genes, which I found in the literature, from the 3 databases that are used to keep nucleotide and amino acid sequences, which are GenBake, ENA, and DDBJ. So as you can see, there is no variation in these files. So these are pretty well documented genes, except for these slightly dated submission dates on ENA, but that's an upload issue, most likely. Has nothing to do with the sequences themselves. So we're looking good. So now we'll talk about blast analysis, and we'll start with BLA-VIM. So here it is. This is a pretty standard blast search. Nothing too surprising. Notice a lot of clinically relevant species like Pseudomonas, Klebsiella, Cyanidobacter, a lot of those escape pathogens that we were talking about, relatively high query coverage and percent identity scores across the board. And notice the volume of results. We'll see we don't have this volume of results with some other genes. So here's the BLAST-P analysis looking at the amino acid sequences. It's more of the same, not much to say. Now for BLAST-PM, you can see already we have far fewer results and lower query coverage and percent identity scores, but we still have some of those clinically relevant species we were talking about, Serratia, Citrobacter. And so here are the BLAST-P results. As you can see, we were only able to find one species on BLAST that has significant homologies to this genes, and it's Acinetobacter baumannii, one of the escape pathogens. Low query coverage scores, but pretty good percent identity scores. So this was interesting. Now onto BLAST QPC. This is more like the Blavier M1s. Nothing much to say about it. You know what to look for. For BLAST-P, fewer results, but what we see is still corroborating the BLAST-N results. So this makes sense, even though we have fewer results and lower query coverage scores. So just a few notable combinations that we pulled out of the BLAST results. There is one species that possesses all of the genes and all of the proteins that we're talking about here, and that is A. baumannii, which is an escape pathogen, not too surprising, we'll talk about it later. And since A. baumannii was the only species other than P. aeruginosa that was found to possess the BLAST-PM protein, I wanted to give note to also some other species that possessed all of the genes and 2 out of the proteins, just not BLAST-PM, and those are Klebsiella pneumoniae, Serratia marchesans, and Escherichia coli. Okay, now to protein analysis. Again, we'll start with Blavier M. So on InterPro, the results support BLAST results for bacterial species, which is good, but we're also seeing some significant homologies in Eukaryota and Archaea, which was surprising. These were absent on BLAST, except for this one eukaryotic species that we couldn't look more into because it had a query coverage score of only 12%. So that means the homology that was found was only on a very little snippet of DNA. We couldn't look more into it. Now this disparity between the BLAST and InterPro results, I guess it's most likely just because BLAST doesn't account for the 3D homologies between proteins. It only looks for homologies between amino acid sequences. So it may be that these eukaryotic and archaic species have significantly different amino acid sequences from the amino acid sequence of the protein in P. aeruginosa, but they might still fold the same and have a similar shape. So the bacteria that we chose to look more into for Blavier M was Bacillus. There was no species specified on the file that was used, but there's a couple different species. It could be a couple different species that we care about. It's gram-positive ubiquitous bacteria, and it has a few clinically relevant species, most notably B. anthracis, which causes the disease anthrax. I'm sure you've heard of it. And now we pulled the Blavier M protein from P. aeruginosa from the Worldwide Protein Databank, or WWPDB, and we predicted the protein in Bacillus using AlphaFold, which is an AI tool. We can see there's this one common domain of mixed alpha helices, I don't know, and beta sheets. And the model confidence for this domain and the Bacillus prediction is very high or high. You can see it's in the blue. But the Bacillus protein also has this second domain, which looks kind of like a tail. But model confidence for this domain is rated very low. So this suggests that it might not exist in reality or that it's different from what's shown here. But overall, they have pretty similar structures, so we would consider this to indicate a shared function between these 2 proteins. Next, we looked at Phobius, which tells us where the proteins are located. These reports, as you can see, look pretty similar. They show a cytoplasmic and non-cytoplasmic component for each protein, so this would support the structure and indicate a shared function. So next, we chose one eukaryotic species from those InterPro results, which is Colectotrichum sublineolum. It's a fungus, and it's a plant pathogen that causes anthracnose and wild rice and sorghum. Now, of course, it's not treated with Carbapenem, so that's why we're interested in why it has this protein. It's usually treated with natural measures, actually, and only occasionally with fungicides. So, same setup, P. aeruginosa on top, prediction, C. sublineolum on the bottom, and we just see this mixed domain again, alpha and beta. And model confidence, Again, very high or high, so we would consider this a shared function, indication of a shared function. On to phobias. So the C. subliniolum report is less similar to the aeruginosa report than the bacillus protein, but considering this alongside the structures, we would say that this supports the possibility of a shared function. Now on to BLASVM. Okay, so like with BLAVIM, interpro results support BLAST results for bacterial species, but not for eukaryotic and archaic species. So there's some surprising results there, and it's probably for the same reason. Now we chose to look at the bacteria Pseudobacteria vorax and Tilogorgi cola. Now we don't know much about this bacteria. It was discovered relatively recently off the coast of Chile. But it's a gram-negative bacteria that lives in marine environments like the sea, and it preys on other gram-negative bacteria. So same setup. We have this mixed alpha-beta domain again, which is again rated very high or high on the P. Tilogorgi cola model. But like bacillus, P. Tilogorgi cola expresses this tail domain, and like in bacillus, it's rated very low. Now, we would say that because in both models, this domain is rated so low, that this would suggest that it doesn't exist in either of the proteins. So this is an odd result, but actually a helpful one for us to determine whether or not this domain exists. Oh, and by the way, we would say that these structures indicate a shared function between these two proteins. They're pretty similar. Okay, phobias, again. So these reports look pretty similar. A mostly non-cytoplasmic location for both proteins that indicates a shared function. But this kind of conflicts with our theory that the tail domain might not really exist based on the similarities between the bacillus and P. Tilogorgi cola proteins, because this phobias report, of course, looks very different from the bacillus protein. So the theory relies on thinking that perhaps the P. Tilogorgi cola and bacillus proteins are similar, but this would question that, put that into question. Okay, so we took another eukaryotic species from interpro results. This time it's Ancylostoma selenicum, which is a parasitic roundworm, more commonly called a hookworm. It infects humans, but also domestic animals, and causes a host of blood-related issues. Again, not treated with carbapenems. It doesn't even have cell wall because it's an animal. It's usually treated with antiparasitics. Okay, now, this prediction looks a little bit different from what we've seen. We still have this mixed alpha-beta domain over there, and we still have the tail that's been showing up a lot. But we have these two notable deviations from the shared domain, which are this alpha helix motif, you can kind of see on the top, and the sort of looping around beta sheet motif that's at the bottom. Now, model confidence for this whole protein prediction ranges from very low to very high, that's less consistent with what we've been seeing. Most of the mixed domain we can see is high or very high. There are parts of it that are in yellow, so they're low. And the model confidence for the tail, again, like we've been seeing, is very low. And parts of these different motifs are low or very low. So they might be different in reality from what they look like here. Or they may not exist. That possibility is always there. But we'll need to look at interpro to determine whether or not the possibility of a shared function is still there. Okay, so here we are, interpro. This settles things. Acylenicum's interpro report is very, very different from P. aeruginosa's and P. Tilogorgi cola's. It shows a cytoplasmic and non-cytoplasmic component for the Acylenicum protein, whereas we saw the P. aeruginosa and P. Tilogorgi cola both had a mostly non-cytoplasmic localization. So this would indicate that the Acylenicum and P. aeruginosa proteins do not share a function. Block KPC, this one will be quicker. So interpro results, corroborated BLAST results completely. This protein doesn't seem to exist beyond bacteria. So we chose to look at Chromobacterium. Again, no species specified. It's a gram-negative bacteria. Probably the most common and well-known species is C. violaceum, which is another ubiquitous bacteria in tropical and subtropical climates. It's an opportunistic pathogen, can cause things like skin lesions and sepsis. And these other species, they're also common. These were found in BLAST results, but they're all just about the same as C. violaceum with variations in pigmentation and things like that. so this time we were able to get both proteins from WWPDB, and as you can see, they look almost exactly the same. There's just this single mixed alpha beta domain with the helixes on the top, helixes on the bottom, and beta sheets in the middle. And this similarity was kind of surprising because this species, Carbapenem, had a relatively low percent identity score, which is only 74.65%. Usually we expect the higher the percent identity, the more similar the protein. So here are the Phobius reports. They're less similar than their structures would suggest. They don't look exactly the same, but these would still suggest a shared function, cytoplasmic and non-cytoplasmic components for both proteins. So, little known on the methodology that can help sort of explain the results. So for the BLAST analyses, all the settings of BLAST were used, and all the similarity species on, all the similarity settings on BLAST-N. What happened was I would take the sequence from P. aeruginosa, the file, and for the first query, I'd exclude no species, but for each subsequent query, I would exclude one or more of the species that showed up most frequently in results. And these were put into big, messy tables that were later consolidated in the tables you saw earlier. The bacterial species that I wanted to look into further, the goal was for them to have relatively high query coverage scores so that the homology was over more than just a tiny little snippet of the protein, or the gene, and relatively low percent identity scores so that there was still a question of whether these proteins really looked the same. Now for interpro, this is really weird and important to note. I took the protein sequences, the amino acid sequences, from the specific species that I wanted to look into further, and I used those for my queries. So all subsequent analysis was based on those specific sequences. If you search for the proteins just by name, and you don't use the specific sequences, you won't find those eukaryotic and archaic species in results, which is interesting. So what all this means. So the problematic combinations that we talked about earlier, they're not super surprising for species like A. baumannii and K. pneumoniae. Those are escape pathogens. They're known to be very resistant to drugs, and they're paid a lot of attention to in clinical settings. But we might want to direct more attention to species like E. coli and S. marchesans that have the potential to cause really dangerous infections, but aren't paid as much attention. We might want to avoid using carbapenems if we can against species that possess all three of these genes and proteins and are really resistant to them, like A. baumannii, and maybe try and use them as a last resort for species that have other combinations. Now for the eukaryotic and archaic species that we found in interpro, now this might just be because the BLAVM, BLASPM genes and their proteins just haven't been studied enough, that there just hasn't been enough empirical research done to yield consistent results with BLAST. And this might, well, it's unlikely to be relevant in a clinical sense because, again, eukaryotic and archaic species are not treated with carbapenems if they cause an infection. So it's unlikely to become a resistance issue. But it's interesting to wonder why they would express these proteins and not have need of them. It might change our perception eventually on the acquisition and use of proteins like this. Now, it might just be that these serve different functions than they do in bacteria. That might be the case. It might be the case for the first eukaryotic species I showed you, or no, the second one, Acelinicum. That might be the case for that protein because as we saw, it was quite different in structure and interpro and localization from the P. aeruginosa protein. But this doesn't work for the other eukaryotic species because we saw the Phobius and structure reports were still relatively similar, so we need to do some more work on that. Some species were listed in BLAST-M, BLAST-P, and vice versa. This might just be, again, due to a lack of data, but if empirical research confirms that those results do exist, then we might want to look into them more. They can raise some interesting questions. So here's what I have as a conclusion. It's just kind of some of the points we talked about wrapped up. I'm not going to read them. We already talked about them, but there it is, condensed, if you'd like. Okay, there are my references all at once. There are many. Okay, thank you. Questions?